We are live on YouTube, guys. Welcome to the cohort call.
Welcome everyone to the Science Cohort public call and this final call of Open Hardware Makers. We're really happy to be here with everyone and with Cecilia, our guest speaker. And we are 2 minutes after the hour, so we're going to start with this call because we have a lot of things to go through. So once more, welcome. And some announcements, as Andre let you know, this call is being streamed via YouTube. So make sure that if you don't want to appear, just turn off your camera. All interactions, as usual, must respect our community guidelines. And if you have any problems or questions about this, you can send them to openhourmakers@gmail.com. So to start, we have a full agenda today. We're going to briefly recap on the science cohort and give you some details about it and how it worked during these last 2 months. We're going to have our guest speaker, Dr. Cecilia Herbert, who will be sharing with us her experience working at OpenEFIS. And we'll have demo time. We'll have videos from all the projects who participated in the science cohort, and we're very excited to hear more about them. We're finally going to wrap up, and we're going to be here for questions if you have any. So to start, sorry, I just have to accept people to the call. This is for people who are following this video on YouTube and may not know what we do, but Open Hardware Makers is an online mentorship program supporting open hardware projects and developers who are mentees in their way towards acquiring best practices, working open with hardware for their project, also building welcoming and inclusive communities, and connecting to existing networks and experts who can help them in this journey. So just a big thank you to our mentors and experts in this cohort who are really the core of our program. We are really grateful to all our mentors and experts. And this science cohort had some special features. So this science cohort, instead of being a 16-week program, as we used to have, was reduced in duration. It's an 8 weeks program. So these were very intensive 2 months of work. We decided to try this format to understand if we could increase engagement and make it easier for people to follow, and also for everyone to get all the necessary details they need to make their project open. It was only dedicated to hardware projects who had an application in science. And one of the reasons for this is because we are very grateful to Code for Science and Society for sponsoring this cohort and supporting us, not only the organizers, but also we were able to support mentors and experts, and also provide funds to mentees for their projects, thanks to the support from Code for Science and Society. And well, it's Code for Science and Society, so that's why this cohort is fully dedicated to science. We had new mentors, we have more experts, and as I was saying, we were able to provide some funding to support the projects. In terms of the curriculum, this time we moved to Moodle, which made things a little easier in terms of communications, and we were all together in one platform. And just to give you a little taste of our curriculum, it's all about best practices working open in hardware. So each week had either a mentor call or a cohort call, and different units that were pinpointing like very specific aspects of open hardware that you need to take into account if you want to open your project. So we are today in the showcase of projects. And I'm gonna move on now to introducing our guest speakers, sorry. Because we're very happy to have her here. We think it's a great complement to our curriculum and to the learnings that we had during this last 2 months. Our guest speaker is Dr. Cecilia Herbert. Cecilia is an Argentine neuroscience, and during her PhD at the University of Buenos Aires, she studied neural coding of song production using extracellular electrophysiology. Her research interests are in behavioral neuroscience and evolved into a passion to teach. So Cecilia has done some amazing workshops on open tools for research. And she took part in many initiatives to bring knowledge about modern tools for neuroscience, such as workshops, talks, courses, with an emphasis on Latin American students. And she continues now to pursue this mission after her PhD at the OpenEFIS production site, which we will hear more about today. So I'm going to stop sharing my presentation now, and I will let Ceci share hers. Perfect, Ceci. Feel free to go ahead.
Hi, everyone. Hi, how are you today? Thank you, Julieta, for that very warm introduction and for introducing the program to me as well. I didn't know this one was focused on science. It's a real honor for me to be here since I have learned so much from both Julieta and Andre about open source hardware, and that's actually how I started. So my roles at OpenEFIS production site are running training, support, and community engagement. And today I wanted to talk a bit about what we do. And you will soon see that it's very much related to what you have been learning about. So we develop and produce and distribute open source hardware and software for research to more than 40 countries around the world. We provide support for those tools and training on how to better use them. And we also do custom designs of hardware as well. Currently, there's a production site in Lisbon and a public benefit company in Atlanta that work together under the same brand to provide these tools and services. So this is the classic acquisition system. I won't get into too many details, but let's take it as an example since it's how this all started. It started with the open source acquisition board for extracellular electrophysiological activity, so that's brain activity. This is the graphical user interface. And this all started around 10 years ago with co-founders Jacob Voits and Josh Siegel at MIT. The board amplifies signals, this whiteboard here, amplifies signals from around 500 electrodes in the animal's brain. And that can be coupled with other hardware for behavioral research and electrophysiological research through other systems that you see here. So during your cohort, you must have learned about the virtuous cycle of open source hardware, probably much better than I know myself. Basically, more than just having access to the designs, having a fully open source design allows you to have documented your hardware, and it's beneficial because that can mean that it's reproducible, it's repairable if it gets broken. You can personalize it to your needs, and that allows for a more collaborative science, which is also typically lower cost. This initiative way back started with all of that in mind, looking to break away from the standard that were monolithic companies that were selling these products and were black boxes for these tools that were meant for research. And the idea was to get a more modular and interoperable system using standardized interfaces that could talk between each other to allow flexibility for researchers. So think about researchers that are pushing the limits of what their hardware can do because they're interested in challenging behavioral questions, for example. And the open hardware here meant that they wouldn't have to let the tools dictate the questions that they can ask. So that is very, very important. And you can read the paper about how these initiatives, the other open source initiatives that were around when this all started, and how really that shaped, for this particular example, which is high channel count electrophysiology, how that really shaped the landscape for open source tools and what we do today. And this was motivated by facilitating the sharing of tools that were developed in each lab and maybe like standalone and floating around, but really how do we actually make better use of that and disseminate it to a wider audience and make them better. So here I've taken this from talk by Jacob Voits. It's the not invented here syndrome, which I think if you have a chance to check out one of his talks, this is very nice concept, which is yeah, you can build something from scratch, but if you take into account all of what's happened in prior projects and you take some time with that, it might seem boring and that you have to understand what those other people were thinking in a different project. But by collaborating with and building off of existing projects, you actually get a much better solution that works better and has more pairs of eyes on it, and adheres to standards and has a whole community around it that build it. So it's beneficial instead of just, okay, working. So how do we do that at OpenIFES? In the case of the open source tools that we produce and distribute, so labs or developers get in contact with us. There's our internal development, which are the tools that I've been talking about up to now. So our classic acquisition system and the next generation acquisition system, also for electrophysiology and other electrophysiological tools. But then, as I was saying, other tools which are not of internal development, we work with labs, our developers, to produce those tools, so to get them ready for manufacturing, review them, quality control them, and then produce them, distribute them, and offer repairs and support. So these includes tools that are, for example, the UCLA Miniscope project, that is for imaging, the FED device from the Kravitz lab for behavioral assays involving feeding, and other tools from the Champalimaud Foundation and HARP platform for other behavioral assays. So much more than just open electrophysiology, but a complete suite of tools and other tools that are not shown here that adhere to this interoperability and, of course, openness. So you will see that what this means for us is that the tools are on our GitHub repository, both the hardware and the software, and the documentation as well, which I will talk about in a minute. And the software is really what is maybe the target of more collaborative development, because in hardware terms for our own stuff, it's a bit more difficult. But for this collaborative cycle, then you have people modifying our software via plugins. And we also make contributions to other software that use our hardware or hardware of this ecosystem. So particular project is the Bonsai project, which is very big, and we contribute packages to that ecosystem of open tools. As for the documentation, of course, here you get also a feed-in from doubts and comments that users have and that we get through our support channels. And ultimately, we try to make those better and better for people to actually use those tools properly and get a maximum benefit out of them. What I was talking about regarding software is very important for interoperability. So I was saying that this is our classic acquisition board, and it works with this GUI, and that is via this plugin for this acquisition board. But that's not only what the GUI can do. So the software actually, works with multiple different hardwares from other manufacturers. So our software has this cross-pollination with many plugins that I was saying that users develop for the software, but also hardware that can be used through our software to acquire other types of neuronal data that our own hardware cannot do. And also our hardware, which is the acquisition board pictured here, can its data can be acquired through this project I was mentioning, Bonsai. So a completely different software that, thanks to the package that is available for our hardware, can run our hardware. So there's this cross-pollination, and then of course the GUI can also interface with multiple other hardware. Bonsai can interface with other hardware, and there's a really lot of cross-pollination between this ecosystem. And towards the wrapping up, I guess one of the biggest areas that we're currently developing is education. So there are different types of educational initiatives that we carry out. One of them would be training, which is one-on-one personalized training with each researcher that bought our tools and bought our training, wants to really get into what is running inside the hardware and how to use it for the benefit of the software for their particular research. Then there's workshops or talks and initiatives such as this one, in which we're broadly and to a more a wider audience than just someone that got our tools in their hands. We're disseminating about what we do and what there is out there for the types of tools that we provide. And then the really interesting one is the acquisition, extracellular acquisition course, which is a course that we run every year about extracellular electrophysiology acquisition. And now it's not just related to a particular product. So this is just theory of what is inside our products. Like ultimately, that's what you see there is inside our products, an amplifier that takes those little voltage deflections and how they are made bigger and how they get to our computer screen into wiggly little lines, right? So students build this in their own hands remotely. They get a kit with these electronic components and they understand the concepts and step by step what is happening in this extracellular electrophysiology acquisition system. And by the end of a week, they get to record their neuronal activity in the muscles and how that gets translated using this software that I was mentioning, Bonsai. So very relevant tools for a modern lab in neuroscience. Yeah. And one thing I would like to mention is that thanks to the Creative Commons license here, there have been derivative works in Spanish of this course material. These materials are online. This is also a course that is certified by the Cajal Advanced Neuroscience Training Program, which aims to give students a broad base on which to then develop their particular research, but basically to graduate students. And as I was saying, the fact that these are open source and we strive to distribute them widely in remote courses, it really means that it has a potential to have a really wide audience, and that's what I'm working towards. Finally, there's a very important community around all of these products and these tools, and there's a community of users that use them, the people that we train, the people that read the papers. We get to interact with them via email, via conferences when we do conferences, and courses that the people involved in the project and in the company are teach at. And then as for resources, you would have the GitHub, of course, our Discord server to communicate between users, social media, and a newsletter that goes out through a Google group. So really, I wanted to say thank you for inviting me, and I hope that this has been informative, and really I can take any questions. You can also write to me or to the company at those emails.
Thank you, Ceci. That was a great overview of what you're doing. And can I ask you a little question? I know there is another question in the chat, and if you want to take that through the chat, that's best, so we can move on to the demos. But I'm aware that you started in this open hardware journey, but on your own, right? With your own project, just as many of the mentees here in this program did. So can you very briefly mention what your project was and why it was important for you to grow into this community?
Yeah, thanks for that. Actually, it's very relevant because I'm starting out in this field. I've only been around here for a couple of years now. Initially, it was by a talk by Andre about open neuroscience and his efforts in Africa. And I was struck by how this can be life-changing for a researcher in an under-resourced lab. And it was very relevant for the types of labs that were around in Buenos Aires at that time. And then I also wanted to teach other students about that this wasn't that hard in a sense, or that it shouldn't be so daunting, that we can sit together and work through what it means to have an amplifier and some custom code to analyze signals and that kind of stuff. So, but I think by mentoring other students and providing these courses, I also started to understand that that was what ultimately I wanted to keep on doing and distribute wider, hopefully to communities that speak English as a second language, and how that could be more accessible to them as well.
Thank you, Ceci. That is super interesting. I'm glad OpenF has got you. They got a great deal. And I think there are some questions, some more tech questions in the chat. If you want to take those in the chat box, that would be great. Or after the demos, we can also do that, as you wish.
As you wish for time, I'm not sure.
Yeah, we can move on now to the project demos, and if you want to meanwhile write down, that works.
Perfect. Awesome.
Thank you,
Thank
Ceci.
you so much.
So there we go. I will share my screen. I will continue with the project demos. And okay, so we are gonna go now with the first of our project demos, which is the project Buffel from Carles. And I'm gonna need a minute to switch to his video. Okay.
And welcome to the final demo presentation of the Baffle project. The Baffle project stands for Bionic Apparatus for Fluorescent Light Estimation, and it was developed at the Systems and Synthetic Biology Laboratory at Center for Genomic Sciences at UNAM University in Mexico. Well, this project was developed as an open hardware optoelectronic device with the aim of acquiring multi-channel time-lapse images of bacterial colonies growing in agar plates and other kinds of biological species. And it was developed using open hardware devices such as the Fidgets, Arduino, the V-slots from OpenViels, the optics from Thorlabs, and it was built using a 3D printer from Ultimaker and a laser cutter from Glowforge. Within this device, the samples are grown under controlled environmental conditions, allowing us to acquire time-lapse images of growing colonies, but also multi-channel images can be acquired in different fluorescent channels, as we see in the photograph for 4 different fluorescent filters. This device also allows us to study bacterial population dynamics in spatially explicit environments. And here, for instance, at the top, we can see an event of plasmid-free segregation bacteria in purple. And also, moreover, we can use image processing techniques and algorithms to determine the spatial distribution of fluorescently tagged strains in polymicrobial communities. This project is targeted for school and academic staff, but also NGOs and scientific staff, and it's meant to be used within high schools, DIY, and do it with other movements, amateur microscopy groups, maker spaces, and biofab labs, and within ecology and bioart collectives. How openness benefits our project? Well, first, the most important thing is that it can be replicated by other people around the globe. For instance, we already began a collaboration with the University of Southampton at the School of Biological Sciences, where they are replicating the device. And moreover, it can be used as an empowering observational tool that can be used from academic and non-academic collectives, and also it can trigger experimental educational practices that can be shaped and tailored within a specific community. So our next step is to become sort of an open hardware evangelist and try to act as a bridge between the technical system from the Baffle and the future users and communities that want to develop the device. And we would like to increase the awareness and improve the perception of the project within the interested communities, and also keep on working with new projects that we have already begun to work in the laboratory, such as the Bactomata, that is an open microscope coupled together with an open trans pipetting robot in order to perform different serial transfers of bacteria and diverse microbiological experiments that we are very happy to share you in the future. And where do we need help? Well, we would like to get our project known within these BioDIY collectives and practices, and we would like to be part of the community here in Latin America, but also worldwide, and be present in gatherings and future meetings. We would like to begin new collaborations with projects or people interested in working together, and also try to exchange practices and maybe residencies here and outside Mexico. So during this course from Open Hardware Makers, one of the most important things that we've learned is the importance of tracing an overall roadmap, but also from the first stages of the project, and how valuable is documenting and having instructions and code repositories and step-by-step how-to guides. And also like the relevance and diversity of all the different kinds of open hardware and open software licenses and standards that there exist out there. And for sure, this course, this whole procedure and educational standard is surely a guide on how to tackle new open hardware projects. So, well, if you want to know more, you can check the article that was already published 1 month ago or so at the Journal of Open Hardware. It's freely available. And here are the references and links for the project, my contact, and well, thank you to everyone.
Okay, so thank you so much to Carles, who I know is in our call. So if you want to unmute yourself and say anything, or if anyone has a question for Carles, then this is the moment.
I have to say something. Oh, sorry.
Yeah.
Come on.
Okay, because hi, I'm Alex. I'm a parental leave OHM team member. Anyway, so when I got into open hardware, actually one of my first projects was a dark field bacteria, also macroscopic movement device. And what I was struggling with it in the end was actually the software side. So my question for you is, first, amazing project. I really loved it. But are you also focusing on software? Are you looking into this as well, or are you just concentrating on the hardware?
Yeah. Well, thanks. We also develop graphical user interface specifically for this device to be controlled. For instance, to set up like the configurations that you want to use. For instance, if you want dark field, then fluorescent in green, and then after 10 hours you want to trigger the red fluorescent filter, and then switch and change the temperature. So it has a lot of different configurations that can be selected within a graphical user interface that we explicitly develop for the project.
Awesome, thanks.
Thank you, Carles. And if anyone has any other question for Carles, feel free to leave them in the chat, or if you are watching this via YouTube, also leave comments or questions there, and we will try to get them through to people. Thank you so much, Carles, for that great demo. And in interest of time, we're gonna move on now to our next demo, which is the Open Ophthalmoscope project. And here we go.
Welcome to our project titled Open Ophthalmoscope. We are PhD students at the University of Tübingen working on interpretable AI model for diabetic retinopathy detection. So the goal of our project is to replicate the open indirect ophthalmoscope, which is a low-cost hardware solution to capture retinal image with an integrated AI software to detect diabetic retinopathy. By replicating this project, we want to update its documentation so that it will be easier for users to use. Now, this project intends to ophthalmologists in general and especially to ophthalmologists in countries with low ophthalmoscope resources to overcome the ear disease screening process. Also intend to researchers like us who use retinal fundus data and by anyone interested by integrating machine learning model into real-world clinical application. Now, why do we work open or how openness benefit to our project? First, we don't reinvent the wheel because we are able to reuse ideas and components from other similar projects. In addition, we can get diverse ideas and perspectives from the community because the project is open. Next, the user have the possibility to send feedback based on their requirement. And finally, we can reach many users by providing a step-by-step documentation. Now, for this project, we have three main communication platforms. The first is a GitHub webpage, which gives an overview of the project to the general public. The second is a GitHub repository, which will be used by anyone who wants to replicate or contribute to the project. And finally, we have a Twitter page, which will be mainly used to update the community about the project.
So coming to the Open Ophthalmoscope project, our main work on it. So we have two main components, the software and the hardware. On the software side, we have a basic app which detects diabetic retinopathy. To this app, a user can upload a retinal fundus image and our machine learning model makes a prediction in the background and prints the prediction whether the image had diabetic retinopathy or not on the screen of the app. On the hardware side, we do not have yet a compact and complete device such as the original open indirect ophthalmoscope project. We are still new to hardware and we are taking a baby steps towards developing a minimum viable project. So in our minimum viable project, we only place a 20D lens close to the eye through which one can see the fundus. And then we try to capture this fundus image with a Raspberry Pi camera. So as said earlier, we are researchers working mostly on developing software tools and we have limited experience with hardware. So it would be great to have contributors from the field of hardware who have more experience with hardware, 3D printing, optics, and product design. These contributors can then bring their ideas and knowledge to this project. And finally, our open hardware mentoring experience. Through the course of these 9 weeks, we have learned a lot of new things. We learned the benefits of openness, how to build open projects. We learned about version control, the process of documenting our project and licensing the different components of our project, and about the minimum viable project, which helped us to break down a complex hardware project into a simpler, more approachable one. We made a web page for our project, which contained user and contributor documentations. We made an app for detecting diabetic retinopathy from fundus images. Going forward, we wish to build a community of contributors for our project. We would also like to improve the hardware and software with contributions from the community. Finally, I would like to thank the organizers, Andre and Julieta, for organizing this wonderful mentoring experience and for all the
great discussions during the cohort calls. We would also like to thank our mentor, Jake Reed, for all the useful discussions without whom we couldn't have come so far. Thank you.
So that was an amazing project from Carol and Indu, who are also in this call, and they have already a question in the chat for them. So either Carol or Indu, if you want to say hi and reply to the question in the chat box. The question is from Sam, and he's saying, really interesting project, guys. How did you train your machine learning model? Sorry, is there a repository of training images that you used? So that's a really interesting question. And we are now going to move forward to our next project. Sorry, I have to change a lot between two project videos, which is the BLE-based indoor navigation system by Benedict Comba. So here we go. Thank you, Benedict, for that, and thank you for your demo of the project. I think it was a little fast. It was a little hard for me to follow. So just FYI, also for people watching us in YouTube, we're going to upload the demo videos to YouTube so you can rewatch and have a more quiet look at everything that happened at Open Hardware Makers. And again, if there is a question for Benedict, who is in the call, just feel free to pop it in the chat. So we are going to go now with our next project, which is the open design of lightweight public constructions. Thank you so much to Natalie and Dimitri. That's a very interesting project. It's something different to what we are used to see. And I know that Dimitri wrote in the chat that they don't have audio, they can't reply to questions, but they are in the chat. So if you have questions for them, I think Sam is asking, please feel free to send them there. Dimitri and Natalie, if you want to write down in the chat your email address, that's easier for people to get in touch. And regarding the previous video, there was a little fast. If we have time at the end, I will try to pass it again in lower speed. But if not, we will always upload it afterwards to our channel. So we're going to move on now to our next project, which is the Startle apparatus from Royhan.
Hi there, my name is Royhan Folari. I'm a lecturer at the Department of Anatomy at Olabisi Onabanjo University in Nigeria, where I also lead the Biopsychiatry and Neuroinnovation Research Unit. So my project, which I am presenting as part of the current cohort of the open hardware makers, is titled the Startle Reflex Apparatus. And this stems from my interest in neuropsychiatric disorders research using animal models, including mice and Drosophila. And it's been known that in neuropsychiatric disorder patients, there is this impaired pre-pulse inhibition, which is a sensory motor gating. This means that the pre-pulse inhibition in them contravenes what is obtainable in normal individuals, where you expect a weaker stimulus to inhibit the level of startle response you get if such weaker stimulus is preceding a bigger stimulus. So when a startle impulse is generated, it leads to a startle reflex, right? And if such a stimulus is preceded by a weaker stimulus, then it inhibits the level of reflex or the startle reflex that is generated. So this is the normal thing in normal individual. But in patients of neuropsychiatric disorders like schizophrenia, there is inhibition of this pre-pulse inhibition. And to understand the disorder better, we always make use of animal models. And this definitely helps us to understand the disorder better and start thinking further means of treatment. So one diagnostic marker to confirm the successful induction of neuropsychiatric disorder in the model, the mice model, is to measure the pre-pulse inhibition as well. And to do this, we need the startle reflex apparatus. And this is where the importance of this apparatus comes to light. So how it works is that there is a weak acoustic impulse that comes before a stronger one. So as is seen here, we would appreciate the fact that at this moment, the animal is there chilling. So once the impulse comes, so it elicits this acoustic startle response, and then the animal gets startled. So the startle is what we're measuring. And once the weak stimulus precedes a stronger one, then we get to measure in terms of the timing of the response. The reactivity score is going to be based on the timing as well as the intensity of the body movement of the animal during the startle. So who are those that find this project, this intervention, who are those that will find it useful? These are the researchers who models neuropsychiatric disorders, especially in rodents or mice. Here is a typical example where you have MK801, which is an NMDA receptor antagonist, disrupting the PPI. So the MK801 is used to induce, is one of those pharmacological agents that is used to induce schizophrenia in rodents. So it disrupts the pre-pulse inhibition here in this animal. So you'll notice the normal animals, they tend to have a higher startle response, okay, while the animals induced with MK801 had markedly reduced level of inhibition. Okay, while for animals that were treated after being induced, treated using clozapine, which is an antipsychotic, we would see that this effect was mitigated. So this is the measure of pre-pulse inhibition, the percentage of pre-pulse inhibition noticed in the animals. So this makes this chamber, this apparatus, very useful to researchers who model neuropsychiatric disorders in rodents. So openness, how does it help in this project? We would see that the use of open source software, open source hardware, open source tool has made it easy to think of fabricating this and to end up fabricating this model. It also encourages or it facilitates reproducibility such that once this project, once this alternative, once this chamber is put together, it makes it easy for researchers to run their experiment in similar ways using this tool. It makes the experiment reproducible, and then it can make us anticipate a similar result from different parts of the world. Openness also has made it easy to afford these apparatus. At the end of the project is going to become much more affordable than what is currently available in the market. And to buttress that point is over here is a commercially available version of the same apparatus. Here is a recent quotation that I received for it, and that's over $8,000. So to be able to make this with way less than $1,000 is going to be a lot of plus to the affordability for researchers who are interested in making use of it. And of course, once it becomes more affordable, then accessibility becomes an advantage. So in the end, all of this go ahead to contribute to the support for neuropsychiatric research using animal models. So openness has added that much value. So the next things now is to conclude the sourcing of components that are needed to build the apparatus as is currently ongoing, then to assemble the components into following the specifications that are currently documented. Then we optimize the apparatus. Then something that we intend to perfect all through all of this is the documentation. So get it well documented for the open source community, the open science community to contribute and to also benefit from. And thereafter, we can do sharing as well and go open with the whole project. So you definitely can contribute in terms of ideas on materials or some alternative materials that could be of help. You also can contribute by helping to experiment the model upon you having access to our protocols. And then we'll look forward to your feedback. That's also going to help a lot in modifying what we already have. So the means of getting across or contribute ranges from the GitHub, Moodle, through different other meetings, and also using our social media handles. So it's been a wonderful experience for me in this, in the OHM, in this cohort, because this is a cohort that it's been the most helpful co-learning cohort I have participated in ever since my first contact with open science through Trend in Africa a couple of years ago. So especially, it's been an idea that I've conceived for quite a while, but being part of this cohort has made it possible for me to get a head start and to start to embark upon the project immediately. So I also enjoyed the peer learning experience through assignments, meetings, periodic meetings where we give updates and all that. So it's been a very wonderful experience for me. And most importantly, this positive energy has been very helpful from the mentors. So I appreciate this with OHM a lot. So with this, I'm able to say that I have, through this cohort, determined and structured my needs for this project effectively. I have commenced the project, and I have also initiated collaboration with specialists in some areas that may be far less than where my background is. So I've had colleagues in the physics department and the electrical engineering department. So going forward, I will continue to see this project to a finish and also share and make open my contribution. So with this, I'd like to thank OHM and everybody who has made this this project a reality. So thank you very much for listening. Bye.
Thank you, Rohan, for the presentation. And we are gonna, I think Rohan is also in this call. I'm not sure, but make sure you leave your questions in the chat, and we are gonna move to our next project, which is the thermal sensor for intradomiciliary monitoring in localities with without reports of the presence of the mosquito, Aedes aegypti. This is a project from Peru. It's in Spanish, but it has subtitles in English, so everyone should be able to share, to follow the video. Thank you, Ceci, so much for joining, and she left there her details if you want to contact her.
Hola a todos, buenos días. Esta es la presentación del proyecto de sensor térmico para el monitoreo intradomiciliario de Aedes aegypti. Este proyecto consiste en un sensor térmico acoplado en un microcontrolador que nos permite registrar la temperatura al interior de la vivienda por un registro, realiza un registro cada hora, es decir, 24 registros al día. El dispositivo ha sido diseñado con la finalidad de, en parte, seguridad para evitar de que los posibles daños y pérdidas, al no ser, digámoslo así, un poco atractivo comercialmente. Aquí tenemos un mapa del nivel nacional, lo que tenemos actualizado, la presencia del vector Aedes aegypti en todo el país, ¿no? De este mapa ahí vemos la región de Moquegua, que es donde hasta el momento no se ha reportado la presencia de este vector. Y luego finalmente tenemos la ciudad de Hilo, que es donde se ha desplegado el proyecto y que, según nuestro modelo estadístico que podemos ver en la imagen del medio, nos indica en el color verde que tiene una idoneidad alta, es decir, el vector puede llegar e instalarse de manera tranquila, ¿no? Añadiendo un poco de contexto, la evolución de la Aedes aegypti, la evolución de la presencia en el Perú es la siguiente, ¿no? El 2005 teníamos 205 distritos y hasta este año tenemos 528, mucho más del doble, y así progresivamente en los últimos años esto ha sido una constante, ¿no? Lo que también se ve reflejado posteriormente en el incremento de casos en los últimos años, casos de dengue. Nuestro país ha vivido grandes emergencias de dengue y estamos todavía manteniendo transmisiones importantes, ¿no? Este año no es la excepción y a pesar de que ya estamos afortunadamente saliendo de la pandemia, este año está siendo bastante más agresivo. Este proyecto podría beneficiar principalmente a entidades gubernamentales, ¿no? Como es nuestro caso. Sin embargo, el espectro de acción puede ser mucho más amplio, incluye entidades privadas e incluso académicas que pueden tener interés en desarrollar este tipo de tecnologías, ¿no? Aquí podemos apreciar el primer mes de seguimiento donde han funcionado los aparatos al interior de la vivienda, ¿no? Nuestra hipótesis es que el interior de la vivienda puede estar más cálido que el exterior, que en este gráfico son las últimas dos líneas verticales, ¿no? Coles y algarrobos. Nuestro plan a futuro consiste en adaptar este dispositivo a un panel solar que permita brindar una mayor autonomía de la batería, lo que nos permitiría asimismo no sobrecargar el trabajo de los equipos locales y también el equipo de mi oficina, ¿no? Esto, a la vez esto podría ayudarnos a incrementar sensores de ser necesario, añadir por ejemplo humedad, ¿no? Que en este momento no estamos registrando. En relación a la mentoría de Open Hardware Makers, ha sido bastante placentera, muy llevadera de cursar, bastante interesante escuchar experiencias. Los organizadores han sido muy amables, ha sido muy grato, la verdad. El- Cualquier colaboración en relación a este tipo de proyectos es bienvenida, ¿no? Sin embargo, nosotros quisiéramos mejorar en cuanto principalmente a la autonomía de la batería o formas de optimizar este diseño. Algo, ¿no? Un proyecto, ya sea el software, el hardware, la guía de usuario, la guía de implementación y todas esas cosas, ¿no? Y gracias también a las conversaciones que pude tener con el mentor que tuve asignado, pude investigar y conocer diferentes proyectos que podrían tener aplicación directa en nuestro trabajo día a día como entidad gubernamental, ¿no? Que podrían elevar el alcance y reducir muchísimo los costos, ¿no? Como es el caso de los colorímetros abiertos, que son muchísimo más económicos. Eso es todo por el proyecto. Muchas gracias.
Okay, so thank you so much, Angelo. And I'm going to move now to our next project. We have only 2 more projects to present. Next project is the mini wind tunnel project from Samuel.
Hey everyone, welcome to the Mini Wind Tunnel project. I'm going to base this talk around the website that I built during the OHM course. I say built, I mean I stole a template from someone else. So this project was born out of the need to address the underrepresentation issue in engineering. So typically in UK engineering universities, the cohorts consist primarily of middle-class white males. And so the idea is to get these tunnels into schools and inspire kids from all sorts of backgrounds to take up engineering higher education. So this wind tunnel is made out of laser cut wood and 3D printed parts, which is really going to help gel with the openness of the project and hopefully lead to a much higher uptake of this project. So I said a little bit about who it's for primarily, but also speaking with some of the open hardware community in my university, and they opened up my eyes to actually having this thing as a legitimate research tool. So that's a really exciting avenue that I want to explore as well. And so openness is the name of the game with this project. The idea is to have as much impact as possible, and if this project wasn't open, we'd be seriously hindering the impact that it could have. So the next steps are to complete the OHM homework, which primarily is the documentation for me. So that's obviously incredibly important for an open hardware project. And so I need to get that done. And I'm going to use Git building for that. So I'll show you a little example in a minute. And where I need help is basically in the kind of coding HTML side of things. So I'm already getting asking questions on this Git building community, and I'm getting answers, which is really, really cool. So it's nice that I feel like I have a footing in the community where I can actually ask questions and get answers. So I learned there's a hell of a lot more to an open hardware project than just simply uploading files to the internet. The OHM has provided this incredible framework to follow, and I'm immensely grateful for that. I made this website and I started making documentation pages. I'm using command prompts, which I usually run away from because they scare me. So that's amazing. So I've got my hardware files linked on the website, and I've got some documentation and instructions as well. It doesn't really work at the moment, and that's what the Git building question was for. But I can show you on my local host kind of what it looks like. So I've been using this Git building framework, which is really, really cool. And what I want to do is actually make a tutorial video for people who are just as hopeless as I am with computers. So yeah, an incredibly powerful bit of kit, and I am just taking it slow to make sure I know how to use this and start it properly so I don't have to redo things later. And so I will continue to write the documentation in the correct way, and I'll continue to follow the framework and best practices that the OHM course has provided. Thank you.
Wow, that was an amazing presentation, Sam, and also great for the Git building project, which is also starting and started as a response to this need for better documenting hardware. Again, feel free to leave your questions for Sam in the chat. And I'm gonna move on now to our last project, which is, it has many names. I think it's the pipetting bot, but I think Nico is in the call, and after the video, he can tell us what's the best name to call it. I think that's not working. Okay, I'm gonna solve that in 1 second. Meanwhile, we remind you, and thank you for those who are following via YouTube, that this was a science cohort supported by Code for Science and Society. And for some reason, I cannot play it, but Nico, I don't know if you want to start sharing it. The file is wrong for me, it's not working. Maybe you have it locally.
There's a YouTube link. I can share it with you if you want.
Yeah, that would be great.
There it goes.
Thank you. All right, should be good to go now.
Hello and welcome to. It is about a pipetting robot mentored by the OHM program. My name is Nicolas Mendes and I will show you what this is about. What you're seeing now is the base plate, which is the surface onto which laboratory objects are placed. The system is based on curves, which are used to align the objects to the machine's X, Y, and Z axis in a simple and reproducible way. The entire base plate can be removed and placed back easily without losing alignment, thanks to a kinematic coupling based on 3 screws on the underside and the V-slots on the aluminum profiles. This system allows you to align just about anything to the workspace, as long as you have a wooden plank and time to code the software interface. What you see now is the graphical user interface. It is a web application served by the same Raspberry Pi that controls and operates the machine. The physical objects placed on the base plate must be reflected here in a virtual workspace. To accomplish this, we define virtual objects called platforms, and each one corresponds to a certain physical object, like a tip box. Once defined and calibrated, platforms can be used across many different workspaces and protocols without need to recalibrate. This means that the researcher can use the tip boxes and tube racks that they already have and not rely on a certain vendor. The pipetting protocols are defined as sequences of simple abstract steps, such as a simple volume transfer between tubes or a waiting period for human intervention. Using tags for tubes will greatly simplify this process, because a volume transfer can be defined between tubes or between groups of tubes sharing the same tag. Before actually showing the robot in action, I'd like to mention two things. First, the Raspberry Pi also serves a Jupyter Lab instance, which can be used to run the protocols defined in a web application or to use scripting to operate the robot. Finally, the Raspberry Pi also serves the full usage, maintenance and assembly documentation made with Git building. This is a liquid handling automation project that aims to put automation on the hands of more researchers so that they can do more science with their time. Openness has benefited this project immensely. Firstly, it has been entirely developed using free software tools. It has grown around the contributions of like-minded people, and I hope that it can be built upon in the same way. One of the great features of this machine, in my opinion, is that it is made from widely available components and many 3D printed parts, all of which were modeled using FreeCAD and are available in the main documentation and in the main repository. The other feature I find amazing is the tool changer. The system is rather common in CNC machines, but this is the first time I've seen one in a pipetting robot. Tool changing provides the versatility and modularity required by the wide diversity of tools and procedures used in science. During OHM, I learned about the high quality standards for open projects, which are easy to forget in the midst of development. Completing the documentation for this first version is the main task ahead. We will then focus our efforts on improving the software and the mechanical aspects of the design. For example, we will switch from GRBL to Clipper and design our own set of open hardware micropipettes. With the valuable help of my mentor Barbora, I made significant progress in cleaning up the project structure and making documentation that others can more easily understand. We want our project to be helpful to others, and for that we need lots of help and contributions. If you are working on a similar project, are interested in interfacing your project with this robot, or simply want to learn more about it, we would love to have a chat with you.
Wow. Well, Nico, you have been receiving comments in the chat already, but Alex, if you want to say anything, I see you smiling a lot.
I can't say much then, that I really like this project and how much effort you put into it and how you highlighted all the different features. I really like the tool changing feature as well. Thank you.
And I think this is our last project. Nico, did you want to say anything? I don't know if you are muted. And Sam, I see a hand.
One last thing I forgot to say. This is like, I don't know, it costs about $250 or $500 to make. It depends on the conversion you make. And that's the only thing I forgot to say, which is, I don't know, like 10 times or 20 times less than other commercial offerings.
Thank you, Nico. Sam, you had a hand up, or was maybe my imagination?
I didn't have my hand up, but I guess I'll just throw my comment here that was really impressive. You must put so much effort into that, and that's quite inspiring. So thanks.
Thank you, and also I think just to close this amazing showcase of projects, we have seen some common features. Some projects are more advanced in the hardware development, others are in the prototyping stage, but I think Sam gave a very nice idea of what OHM is trying to do, which is trying to provide a framework so people can better understand how to work on it. So I don't know if before closing this cohort call, first of all, I want to congratulate you all because the projects are amazing. So big congratulations to everyone. Lots of hard work in this compressed version of Open Hardware Makers. You should be super proud of yourselves. Thank you so much to our mentors and experts who I know some of them were in this call and some of them are in this call today. Alex, Andre, if you want to say anything before we close the call. I just can, well, repeat what you say, so I don't do it, but amazing project, amazing projects, and I really enjoyed it. Thank you all. Yes, thank you all. And just be aware that because with this cohort is coming to an end, it doesn't mean that we disappear from here. So we are trying to grow this community of OHM alumni, and you are welcome to send us emails and contact anyone, any of the experts if you want to, and let us know if we can help our project in any way. So thank you so much for participating in OHM. Thank you to our mentors, to our experts, to our fellow organizers, Alex, who was on paternity leave, and Andre, who is in Africa, I think, right now, doing workshops. So thank you, everyone. We hope you have a great project life ahead, and let us know how it goes.
Thank you. Really appreciate it.
Thank you. And I think I can't stop the streaming. I think it has to be Andrea. Yeah, but I'm not sure if he's around. Thank you, everyone.
Thank you, everyone. I think we just leave.
I think we can just leave the call, right? Okay. Bye, everyone. Thank you for joining.
Bye. Thank you. Thank you, Julio. Thank you,
Goodbye.
Alex, and thank you, Andre.
Bye.
Bye-bye.